As John said, I've been in this for a while, and I just want to start with this first sentence because this is how it's going to end as well. If we want wild salmon, it is up to us. And this is a story of why I feel that way. In 1984, I found the perfect place to be, to do research. I was at the time studying killer whales. I wanted to learn what they were talking about. And so I followed them into this place in October of 1984. It's the archipelago between Kinkham Inlet and Knight Inlet. There's a little community there called Echo Bay. And so it was just what my husband and I and baby needed, a little town with a school, and there's no roads, you have to get there by boat. There's no electricity, you have to make it yourself. And I stayed there for 26 years because first, it was the perfect place to study whales, and then it became home. When the first salmon feedlot was towed into this area in about 1988, we thought it was a good idea. Thought it would keep the school open and take pressure off the wild fish. And as I ran around the inlets in my speedboat, I looked forward to these places being shelter from the storm if my boat broke down. But they were the storm. They became the storm. We were definitely wrong. The government still tells us this is good for us, but there's only 8 people left in Echo Bay. The school's closed, and there's 27 Norwegian-owned feedlots. Salmon feedlots break the natural laws that protect wild salmon from disease. A wild salmon moves all the time once it enters the ocean. But the farms hold them stationary. Salmon travel thousands and thousands of kilometers. And so their waste is spread throughout the ocean. There's also a predator, or more than one, for every life stage of salmon. So from the moment the egg leaves the mother's body, if it's not properly buried in the gravel, the water oozels and the trout eat them. And then when they're the little fry, it's the kingfishers and the mergansers, and then on up the food chain until finally it's the grizzly bears, the orcas, and us. But the salmon farms are net pens in the ocean, and up to 1 million fish will stay in one place and defecate into the water right there. The sick fish aren't taken out by predators. In fact, quite often they're given drugs to keep them alive, but in still a contagious state. And they're parked on the migration route of the wild salmon. So in the fall, during the Cohen inquiry, one of the presentations that was made to us was by Dr. Kyle Garver of DFO. And he said that during a disease outbreak, each salmon feedlot can shed 60 billion viral particles per hour. And there's 127 of these farms located throughout the migration routes. One of the things that's most extraordinary to me about wild salmon is that all the adults that come into the rivers in the fall die. You'd think an animal that had gone out in the sea and fed and come all the way home to the exact place where it was spawned and grew up as a little egg, you think nature would send it back out again to do it again. But instead, the fish dies. All of the Pacific fish do this. They leave the wealth of their bodies to the juveniles. And one of the things that happens as well is they break the cycle of disease by doing this. So that all winter long, there's not huge populations of wild salmon close to the shorelines, in the inshore waters, in the inlets. And the tides wash the coast clean all winter. And so when the juveniles hatch in the spring and eventually come out of the gravel and travel down the rivers, they don't meet the pathogens of their parents until they're much larger themselves. It's quite an extraordinary system, very, very resilient. The fish live in all kinds of different rivers, survive the changes of their rivers. And so I think we need to look at that system as being perfect. And we just need to figure out how to get out of its way and allow it to keep functioning the way it has done. But today, millions of salmon circle in feedlots where there should be none. The viruses, sea lice, breed and multiply in places that should be clean. And it would be really foolish to expect the wild salmon to survive the viruses pouring out of these feedlots. to survive a change in their ecosystem, in their natural history, the presence of a new species of salmon doing something very different from them. It would be foolish to think they could just handle that and carry on with no impact whatsoever. Most feedlots shovel the manure that falls to the ground. But that's not the case with the salmon farms. They never shovel their manure. The reason they like those net pens, one of the reasons is because all the waste just goes to the seafloor, and it's a lot of waste. 5 to 7 tons go in per day, and what goes in, most of that comes back out again, along with mucus being shed as the fish are breathing and pathogens being shed. This waste changes the growth of the seaweeds. The flatfish that live on the seafloor are on a very different bottom than they would be in a natural situation. And they have diseases I haven't figured out yet. No one seems to know what they are. I've sampled fish like this many times, and every time I send them to the labs, they tell me the samples are rotten, even though they're coming from very, very fresh fish. So I still don't know what this disease is, but very common. Also, by putting so much nutrients into the water, it's turning colors that the elders of the communities I've been in have never seen. Atlantic salmon eat fish, and here you can see a salmon smolt in the stomach of this fish. It remains unknown how much wild fish this industry is harvesting as wild fish enter the pens, attracted by the lights and also the scent of the feed, and are eaten by the farm salmon, because we're not allowed to look in the pens. I've talked to the farmers about this a lot. I've asked to just be on the processing lines. When they take the guts out of the fish, I just want to look at the guts, but they won't let you look. So we all know what that means. If somebody doesn't want you to look, it's generally because they have something they're hiding. Blood water pours from the fish farm processing plants. This is right in Discovery Passage, down 90 feet. It looks black in this picture because the light's so scarce down there, but this is red. And it's pouring right into the largest salmon migration route we have in British Columbia. The processing plants for terrestrial mammals aren't allowed to do this, but government turns a blind eye when it comes to salmon farms. These are hatching sea lice that came from that blood water. A friend went down and just held a plankton net over the end of that pipe, and he put the contents in jars for me and put it in ice and put it on the bus, and I got it. And by the time I got it, the sea lice eggs were hatching. So if the lice were alive in that blood water, the viruses and bacteria very likely were also viable. We don't know how to turn these viruses off. The sea lice you can turn off. You just take away the host fish that they're residing in, and the lice will go back to their normal pattern. But viruses are a little bit more tricky. We don't know what fish they can infect. We don't know exactly what they can do. They're very capable of mutating. They're extraordinary, successful organisms. So my sense is not to play around with these things. For years, I thought viruses were out of my ability to track, and I concentrated on sea lice. Sea lice were very interesting to study because they're like a virus with a timer on it. So they get on a fish and then they change their body shape every few days for the first 30 days, and it becomes very clear where the lice are getting on the fish. It's a little bit more difficult with the viruses. Now, we were very fortunate to have the Cohen Commission into the decline of Fraser sockeye. Stephen Harper called this commission at the end of 2009, end of 2009, when the Fraser sockeye collapsed so astonishingly that he felt, I guess, that something needed to be done. And what it consisted of was a number of teams that were put together. There was First Nation teams, commercial fishermen, environmental teams. Nobody wanted to be on my team, so it was just me. And we all got lawyers, and the commission paid for those lawyers. And then the commission, Justice Cohen, called for government to submit all the documents that they thought could possibly have to do with the decline of the Fraser sockeye. And that resulted in 500,000 documents going into a legal database. And as a participant, I was given the passwords to that. And at first I thought there would be nothing in there, but my lawyer said, "Are you kidding? 500,000 documents? You go in there and look." I spent 9 months at my computer because for 20 years I had been talking to DFO about where they were putting the fish farms, about the algae blooms, about the whales being displaced, about sores coming on our salmon, the sea lice. And when I got the keys to those documents, suddenly I was on the inside reading their emails about all of this, about me, about the questions I asked and other people asked. And I learned a lot. Government knows more than they're saying. In fact, their own scientists has figured out what's happening to the wild salmon on the south coast of BC. Crucial information is being suppressed, hidden, and ignored. And this talk is about the viruses. There's now 3. When I first made this talk, there was 2. I had to edit the show just at the last minute. I'm very thankful that you did that. Because every time you look, it seems we find another one. So we're going to start with the first one. In terms of the Fraser sockeye, there's only one thing that explains why the Fraser sockeye began vanishing in the 1990s, why only certain runs of the sockeye vanished, what was found in them, why they returned in 2010, and perhaps why the Cultus Lake sockeye were the hardest hit. And here's what government didn't want us to know. First of all, the timing of it. Here's a graph of productivity produced by Mike LaPointe, Pacific Salmon Commission. So productivity is the number of adults coming back from each set of adults that spawned. That's where the salmon farms arrived. So this is just one graph, one correlation. All by itself it means nothing, but it is rather dramatic. So then if you look at what runs were affected, the orange bubbles are showing how much below average the returns were, and the green bubbles are showing how much above average the runs were returning. We've got a lot going down, one that's going up, and this pattern starts in the early 1990s. Pre-spawn mortality was affecting a lot of those orange bubbled fish. So pre-spawn mortality is fish sockeye that make it all the way back to the spawning grounds and then they die with the eggs or the sperm still in them. It's a real tragedy. The fish has done the whole trip and then is unable to spawn. This was a huge problem that we didn't really hear that much about. By 2001, en route mortality, which is dying in the river before getting to the spawning ground at some point or on the spawning ground. By 2001, en route mortality was as high as 90% in some stocks, and pre-spawning mortality ranged from 10 to 30%. So of the 100% that enter the river, 90% are dying on the way, and then the ones that actually make it to the spawning grounds, another 10 to 30% were dying. Prior to 1995, the total freshwater mortality, so en route and pre-spawn, rarely exceeded 20%. Using a very conservative estimate on the ex-vessel price of $10 per fish, the losses just to fishermen associated with this problem likely exceeded $72 million in 2002 alone. And this occurred year after year. I just wanted to make this point again because as the commercial fishermen were being cut back, the fish did not respond. If they had been the problem, the theory is the fish would have started to recover, but they didn't. So as I'm reading through these Cohen documents, I found a group of biologists within DFO who were extremely concerned. The fish were entering the river early, they were dying many things, nothing stood out as a cause. They thought it was high water temperature for a while, but some of them were saying, you know, it can't be high water temperature because we're losing the fish in cool temperatures and warm temperatures. They thought it was a parasite called Parvicapsula, but the same thing, it was not tightly correlated to the fish that were dying. And here's what some of their emails said. Bleeders, they called them. These were fish, they just touched them and they started bleeding. They did a beach stain, the fish just instantly started bleeding. Definitely something out of the ordinary. Possibly the worst looking gills I've ever seen. Lesions are unique, severe inflammation, mystery deepens. gill pathology is profound and highly unusual. Finding everything but the kitchen sink. That's by Christine McWilliams. She's a vet with DFO. She's generally very precise in her language, but she couldn't figure this out. She'd look at 20 fish from the same spawning grounds, all died with their eggs in them, of different things. And this just didn't make sense to anybody. DFO did not support these scientists. They complained of not having money for couriers just to move the samples down the Fraser system to the labs. But they themselves became so interested. I think some of them were just doing it on their own time. Now, generally, when you lose a fish stock, you don't get to look at it because it's lost somewhere in the ocean. But these fish were unique that way because they were dying right on the riverbank. So in 2006, DFO finally realized that they couldn't do a test set in Johnson Straits and say, okay, we've got the right composition of stocks, we can open a fishery, because they didn't know if those fish were going to survive to spawn or not. So they went to Dr. Christy Miller at her laboratory. She's a DFO scientist at the Pacific Biological Station in Nanaimo. And they asked her if she could predict how many of these fish were going to survive to spawn. And she and her lab use a remarkable technique called genomic profiling. So she's not actually looking at diseases in the fish. She's looking at switches in the cells that turn on and off in response to all kinds of things. All living things have this. And so if a fish has been starving or encountered a toxic algae bloom or sea lice or bacteria, different patterns of switches go on and off. When she read the patterns that she got back, first of all, she could predict how many were going to live or die. So when she took a sample in Johnson Straits, what they did was quite remarkable. They seined the fish, and then they tagged them with a little acoustic tag so they could follow them. And they took a little non-lethal snip of gills and let them all go. And what she found out was the ones who lived had one set of switches on and off, and the ones that died, the pattern was completely different. So the genomic profiles were very different for these two fish. And when she looked at what switches were flicked in the dying fish, it was retrovirus, leukemia, brain tumor, cancer, and she just started to look around. Well, what virus fits this? And her compass went straight to salmon leukemia. This remains a work in progress. So here you see the two different profiles of the fish. And she said the dying sake all had the same pattern. It was consistent with a retroviral infection, a class of viruses that often causes cancer. Now, I should say, and she feels a little bit exposed because in what I was reading was her thought process, her early PowerPoints, her briefings, her funding proposals. And so some of the things that she originally thought, she's now changed and modified. But other things, the pattern has just become stronger. And so this was when I found this document, and I spent days trying to figure this one out, because I wasn't allowed to talk to her. I wasn't allowed to ask questions. I wasn't allowed to tell anybody I had seen these documents. But this was a PowerPoint and it's now public, where she says an epidemic of novel cancer-causing viral disease may be associated with wild salmon declines in BC. Pretty shocking. DFO knew all about salmon leukemia because in the 1990s, they wrote 8 scientific papers on them. And they called it plasmacytoid leukemia, and they said the causative agent was salmon leukemia virus. They named it and measured it. They said it's the size of a virus, it behaves like a virus, it infects like a virus. They called it a retrovirus because of its behavior. This work was done by Michael Kent, Dr. Michael Kent. He's no longer with DFO, he's in Oregon now, but he was then the director of the Pacific Biological Station. It started in 1988 in Seashell. Nobody had really seen it before that, in Chinook salmon farms. It was a new disease. And then when they decided Seashell Inlet wasn't such a good place for salmon farms because of the toxic algae blooms, the industry got up and moved to the Discovery Islands off Campbell River, and the virus followed them. At some farms, it killed nearly 100% of the Chinook salmon in the farms. It was the primary reason for financial failure of many of the farms. The potential for epidemics of an infectious cancer raised the interest of the public. Yeah, definitely, because they're raising food. These fish were for people to eat. And concerns over public health and food safety coupled with a perceived threat to wild salmon health. So all of these are quotes from papers. There's one PhD thesis by Dr. Craig Stephen that was very helpful. He spoke a lot about this disease. And from that document are these statements. The hypothesis that marine anemia is a spreading infectious neoplastic, meaning tumor-causing, disease could have profound regulatory effects on the salmon farming industry. But those profound regulatory effects never happened. DFO just closed their eyes to this. So this is Vancouver Island. There's the salmon farms and the mainland. And these are the Discovery Islands area, which for the Fraser sockeye are the most narrow passages in their marine migratory corridor. And as Dr. Kyle Garver said, when there's an epidemic, 60 billion viral particles per hour can come out of these farms. And when he modeled it, it looked something like this, with the viruses spreading the entire width of the channels. So there's no way for the fish to get through those channels without breathing in these viruses. And here come the sockeye and other fish, and they pass through that, and they've all been exposed. It's like running sheep through sheep dip, but the opposite, where instead of trying to kill something, you're actually, trying to kill a parasite, you're actually loading it with viruses, probably bacteria as well, sea lice. The way fish breathe, of course, is they take water in, they pass it over the gills. So these viruses are coming into direct contact with the bloodstream of these fish. DFO did experiments with this disease. So it has many names. They call it marine anemia, plasmacytoid leukemia, salmon leukemia virus. And what they found is that after 10 weeks of exposure, all of the sockeye and Chinook salmon became infected. And 100% of the sockeye exposed to this disease died. Most of the Chinook exposed to it died. Strangely, coincidentally, Michael Kent was chosen by the Cohen Commission to write the report on the potential for disease, in particular viruses, to be harming the Fraser sockeye. He did put a passage in there about salmon leukemia virus, so he is the primary author of most of the papers, most of the research done on it. And he says the disease causes severe anemia and is usually lethal. But he put it as low risk because it's not natural to sockeye salmon. But his own work found 100% of the fish became infected and died when they were exposed to it. So the first generation of Fraser sockeye that swam through that came back, and that's when the decline began. Dr. Miller goes looking at those fish that are dying mysteriously in the river as she finds a signal, and the only thing that fits it is that virus. How does that add up to low risk? It doesn't. Now, DFO unfortunately never finished their work. They never actually visualized the virus. They never sequenced it. They never cultured it. They only said, we think it's a retrovirus. We're going to name it salmon leukemia virus. But now, today, because they never finished the process, we can't track backwards. We can't look at those Fraser sockeye and say, yep, it's salmon leukemia virus. So that has greatly handicapped us. But the other factor is there's one group of Fraser sockeye that do not appear to go north. Their DNA is never found in samples along the eastern coast of Vancouver Island. They're found on the west coast, first thing in the fall. And these are the Harrison sockeye. And as you can see from the graphs here, over the same time period, those fish have been increasing. They don't pass the salmon farms. Dr. Miller looked at 156 of them, and she could not find this genomic profile of the sick fish. They have another life history strategy that makes them different from the rest of the sockeye, most of them anyway, is they leave the river right away. And so that means after they hatch, they head out in the spring before everybody else who's come down the salmon farm route goes into the river. So they miss the exposure to the adults, and they also miss the farms altogether. The Pacific Fishery Resource Conservation Council called them British Columbia's first and only salmon stronghold, and they don't migrate past salmon farms. Christy Miller, I have enormous respect for her. I have never encountered anybody like her in DFO before. She was very careful in her early work not to mention aquaculture. She said in some of her emails, we have to be careful about this. But finally, in April 2010, she said, I want to test some farm salmon. I want to look for this genomic profile. I want to look for salmon leukemia virus. And she was denied access. And she still is not allowed to test the farm salmon. When 10 million Fraser sockeye went missing, the Privy Council of Canada muzzled her and said she was not allowed to speak to the media. She published a paper in the Journal of Science shortly after. The media was clamoring to speak to her because a mystery leukemia virus in the Fraser sockeye causing the decline after we have a $28 million commission to answer that very question. You can bet the media was interested, but she was not allowed to talk to them. I attended a meeting at Simon Fraser University after the collapse. We talked about squid and temperature and plankton, and we had no idea, I had no idea, some of the people in the room did, but I had no idea about her work. It was completely hidden from us. And so we made a public presentation and said to the public, we don't know. There's really no evidence. Maybe it's climate change, but it's not really fitting the pattern. Maybe there was a plankton problem that year, but it didn't explain the whole 19-year decline. And so the public was misinformed. The fish are talking, if only we could hear them. If you look at them closely, and the genomic profiling is a phenomenally powerful tool. If we just were able to use that in an unfettered manner, we could figure this out very, very quickly. It's a great theory, right? Farms go in, the fish crash. But what happened in 2010? And I became very curious about that when I had access to all these databases. And one of the databases I had access to was the stocking data. So every single farm back to about 2004, up to present, I had the data on how they were stocked. And I took a close look. So for the whole Fraser sockeye migration route inside of Vancouver Island, these were the numbers from 2002 forward. In 2008, there were no Chinook farms on the Fraser sockeye migration route. The farms that had salmon leukemia that were hosting this disease, the record of the virus was still in the provincial audit records. When those farms were removed, the first generation that went to sea without those farms came back in 2010. So now we have more than just one correlation. We have a lot of evidence. And that was the productivity of the 2010s. So it does make you wonder whether these fish were trying to come home to us every year, if there really was no problem. When I sat in the meeting in Simon Fraser University in 2009, the scientists said, we got to face, maybe the Fraser River just can't make sockeye anymore. Maybe it's too warm. Maybe there's not enough food out there. And they were very demoralized by that. The men and women who count them and try to forecast them, they're embarrassed because Alaska's still forecasting their sockeye okay, but they're not. And that would suggest, as a variable that has a very strong effect on the populations of these fish, that they're just not factoring in. And I would say it's probably the mortality rates of these viruses. And there's one more thing you should know about this disease. One of the symptoms that was described in the early DFO work was tumors right behind the eyeballs in these fish. Some of them were sent to the Smithsonian Registry of Tumors. And so Miller, being very curious about this disease, began to look in the brains of these fish, and sure enough, she found tumors. Now, once she found this, all hell broke loose for her, and she's had to withdraw this whole line of work. But these were not caused by blunt trauma, which is what the province tried to tell her. And I couldn't help myself. I phoned her and I said, so just tell me, you wanted to send this brain in for structural analysis, which histology is. You wanted to see, how the shape of this brain was, and you hit it with a stick first. And she said, no. Those fish had not been hit over the head. There was no blunt trauma. These growths had blood vessels attached to the brain. So, of course, this is a concern for the First Nations of the Fraser River. They would rather not be eating something that has tumors in it. When I read about the draconian Chinook cuts looming for anglers, I wonder if these fish are also being affected by the various viruses from the farms. Now, so that's kind of where we were at when the Cohen inquiry closed. But then ISA virus became apparent. I saw over 1,000 mentions in the provincial audits. So a vet 4 times a year looked at several hundred farm salmon, and he recorded over 1,000 times the classic symptoms of the infectious salmon anemia virus. But when he was asked on the stand about that, he said, well, kind of looked like it, but it wasn't. We did the tests, we did the PCRs on them. It was not ISA. I wondered about that, but I couldn't really go anywhere with it. He said that wasn't the problem. There was a lot of questioning. I asked my lawyer, make sure these guys get questioned about ISA. So Christine McWilliams, the vet with DFO, said there's been no indication of ISA or ISA virus. Dr. Kent said ISA virus has not been seen in BC. Dr. Gary Marty, the provincial vet, said he has a great deal of confidence that we don't have ISA virus in British Columbia. Claire Backman, who's with Marine Harvest, said the level of surveys done in the country of origin and then again the quarantine and follow-up sampling here in British Columbia have been successful in preventing any exotic disease, including this particular one, ISA virus. Peter McKenzie with CIRMAC, we've done over 5,000 tests for ISA virus. They're all negative, and that gives us extremely high level of confidence that our industry is free from ISA virus. But there's no reason that ISA virus would not be here. It was never listed on the forms. So every time a hatchery from Washington State or Scotland or Iceland sent eggs into British Columbia, somebody had to sign a form. There was diseases listed on that form, but not ISA. So no one had to put the signature to say, these eggs don't have ISA. It's reportable internationally, but it was not reportable in Canada. So I don't really know how that worked, but there were no reports made. In 2004, the fish health regulations of Canada were waived completely to permit Atlantic egg import from a non-compliant hatchery in Iceland. And the BC audits report over 1,000 classic signs. Every line of defense was lowered. So a friend of mine, Dr. Rick Rutledge from Simon Fraser University, is up in Rivers Inlet, and he's studying the Rivers Inlet sockeye, which have a remarkable similar pattern of decline. And he's trying to figure out why. And last spring, there were so few outgoing smolts that he phoned me up and he goes, Alex, you got any suggestions what we could test these fish for? I couldn't tell him what I was reading in the Cohen inquiry, but I said, test them for ISA. And he threw them in the freezer and didn't really think much about it, sent them off in October, which is the wrong way to handle a fish if you're going to look for viruses. He was really storing them for stomach content analysis. Sent them off to the lab, boom, boom, ISA virus, European strain. I cried. It hit so hard because this virus started in 1984 in Norway. It spread all over the world. In Chile, it mutated into such a virulent form that it caused $2 billion worth of damage. You read the history of the virus in Chile, and they said they had no idea it could spread so fast or kill so many fish. So we held a press conference because we thought, okay, let's let the scientific community know. We sent it to the world. There's only 2 labs in the world that have been certified for ISA virus detection by the OIE. The OIE is the animal health portion of the World Health Organization. And it's in Prince Edward Island. So this is a top lab. This is the lab that diagnosed the disease in Chile. We had no reason not to believe him. Still don't have any reason not to believe him. And boy, all hell broke loose when we did that. The CFIA confiscated our samples. They went into my friend Nissa Reed's house, took them out of her freezer, what we had left, went into Dr. Kabenge's lab, took them. Miller also had some of them, went to her lab, got those samples. Never have returned any of those, even though they've told us ISA is not in them. The BC Minister of Agriculture said reckless allegations based on incomplete science can be devastating. I have personally asked Premier Clark to reassure our valued trading partners that now, as always, BC can be relied on as a supplier of safe, sustainable seafood. So she's on a mission to China, and he thinks that he has to tell her that we don't have ISA there. I thought that was very interesting. The Minister of Fisheries said we had needlessly damaged Canada's reputation. So the only thing I know to do is just go out and start looking. I did this with sea lice, done it with lots of different issues on this. So my friend Anissa and I, we went down to the Fraser River. There were some really friendly people there with a boat, took us out. We began to net fish that were floating dead on the surface of the river. One of them was this 25-pound spring salmon from the Harrison River. It was strangely yellow. It had ISA. This Fraser sockeye had ISA, and this coho had ISA in the heart. And we only took 11 samples. 3 of them had ISA. I cried again. Didn't help. None of them had spawned. And then Justice Cohen did a remarkable thing. He reopened the Cohen inquiry. Because this disease is reportable to the international community, because it has such a lethal reputation, and because it's spreading with salmon farms, he reopened the inquiry and he brought together all the scientists who were looking at it. So on the left, in the front there is Dr. Fred Kabenge from the University of Prince Edward Island. In the middle is Dr. Christy Miller. And on the right is Dr. Nell Gagne from the DFO lab in Moncton. And up above on the screen from Norway Live was Dr. R. Neeland. Because as soon as my friend got positives in those Rivers Inlet sockeye, I tracked down those fish, took out their gills, and sent them straight to Norway. Because I knew things were going to go sideways here, and I wanted somebody in another country to have samples of them. And all of these doctors found ISA in our samples, in other samples, except Nell Gagne, who works with DFO. She couldn't find it. And as these scientists talked, we learned why. She uses a machine that is not good at detecting ISA when it's at low levels, which is what all of these fish had. DFO was quick to say, well, if it was low levels, all these fish were healthy. But Miller's genomic work, the switches that are turned on and off in those fish all indicate the fish is fighting the flu. ISA virus is a flu virus. It's of the influenza family. Dr. R. Neeland said, oh yeah, 80 to 90% of Atlantic salmon test positive for ISA virus at egg take. 30 million Atlantic salmon eggs have entered BC since 1986. So if you want to believe that ISA virus is not in British Columbia, I feel you might as well believe in the tooth fairy. There was no screening for it. You didn't have to sign the form. You didn't have to report it. It's in 80, 90% of the brood stock. Of course it's here. And then the real bomb dropped. Back in 2002 and 2003, Dr. Kabenge's wife, Molly Kabenge, with Dr. Simon Jones of the Pacific Biological Station, a parasitologist, had done research on ISA, and they found it in 100% of the cultist sockeye that they tested. DFO hid those results from the Cohen inquiry and from the Stolo First Nations, where those samples came from. And here's a graph or a chart from that. 64 out of 64 tested positive for ISA. Now, they gave us a lot of reasons as to why they don't believe this test, but they never followed up on any further testing. The Cultus Lake sockeye are the most endangered Fraser sockeye population. There is a Cultus Lake sockeye recovery program underway. And DFO did not think it was worth giving this paper to Justice Cohen, putting into the inquiry. I hunted for any positives for ISA. I felt certain there must be some in there, and I couldn't find them, and that's because they'd been hidden. So a little bit about the cultists. 100% of them died in 1999, 2000, and 2001. They're of acute concern since 1996 due to entering the river too early, pre-spawn mortality, en route losses. 2006, 85% of the cultist lake sockeye died before spawning. Only 2 out of 85 females spawned. Very disturbing. This is a very damaged run. You find the most lethal virus known to salmon in it. Certainly that was reportable to the Cohen Inquiry. Justice Cohen also called the Canadian Food Inspection Agency, and Dr. Kim Plantons got up and she said, she really gave us, you know, we saw what they did, but the question was why, and she really gave us why. She said, so if, let's say, we do find ISA in BC and all of a sudden the markets are closed, our role is then to try and renegotiate or negotiate market access to those countries. We'll let them know what we can do and whether we can meet that market access. If we can't meet it, then there will be no trade, basically. This is a theme that kept coming up and coming up. Trade sanctions, trade this, trade that. She said that if ISA is in BC, that trade might end. Well, ISA is in British Columbia. So the trade partners have been misinformed. So this really piqued our curiosity, and the fish farmers are very possessive about their fish, won't let you test them, won't let DFO in there, won't let any university in there. So we went shopping. They gotta let these fish go at some point because they're trying to sell them to people. What did we do the first? I think we did 11 the first time, and this one was positive, that one was positive, that one was positive, that one was positive, and we threw in some sockeye or some chum from the Vedder River, and this female was positive for ISA. So it's in the supermarket. We now know that Loblaws, who own the supermarket chain where we got these fish from, they said these fish were raised in British Columbia. So we've got the Dr. Gary Marty's been doing the audits. He's saying he couldn't find it, or he thought he saw it, but he said it really wasn't ISA. We've got Cermak, Mainstream, saying they tested 5,000 fish, they couldn't find it. We have Marine Harvest saying, oh no, they're definitely sure they don't have it. But we go to the supermarket and boom, 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 on one day, we get it in 4 different markets. ISA virus is here. Not only did they find ISA virus, we just found out recently in another batch that we have 2 really virulent mutations, one of which caused the massive outbreak in Chile and is considered the most lethal ISA virus mutation. That's called HPR7b. The lab also reports we have Piscine Rio virus, heart and skeleton muscle inflammation. This is a virus. When I sent the samples into the lab, I just thought, well, while we're getting tested, I'll throw this one in. I'd read a little bit about it. DFO and the province said we didn't have it. They didn't get mentioned at the Cohen Inquiry. But it's a nasty heart virus, and it's from 1999 through 2010. It spread through 400 farms in Norway. It weakens their heart muscle. If the potential hosts are in close proximity, it goes through them like wildfire. So they're saying that if there's fish outside of the pens, it spreads very, very rapidly. Several of the articles warn against this. You should try to contain it. This is one of the fish from Superstore that had it. Didn't have ISA, but had heart and skeleton muscle inflammation virus, the Piscine Rio virus. Imagine you've got a weakened heart as a salmon, and you've got to swim against Hell's Gate. How's that going to go for you? Or you've got to evade a killer whale coming after you. That's going to be a little bit tricky with a weakened heart. So is catching a herring. They describe the heart affected by this virus as soft and flabby. The farm salmon often recover from the virus, and you're going to hear that probably a lot, that the fish recover and only 10 to 20% die. But while the farm salmon are recovering from this virus, they're lying up against the net, swimming abnormally. That is not an opportunity that the wild salmon has. They can't rest. There's no place. There's no infirmary. There's no place for them to go and safely rest and have food fall from the sky. These viruses, I call them the apocalypse of viruses. The three viruses now that we know are in these fish, we know they're lethal, we know they come from salmon farms, we know they're spreading. The people who are forecasting wild salmon are going to have to see them and admit to them and actually use them to try to figure out how many fish are going to come back. One of the papers talking about this heart and skeleton muscle inflammation said, at a time when humans are being encouraged to eat fish to help combat a range of conditions, including coronary disease, it seems somewhat ironic that heart disease seems to be such a problem in the fish themselves. Government has failed us. They're asleep at the wheel or worse. These farm salmon will never replace the wild salmon. The wild salmon build the forest floor. They feed the trees that make the oxygen we breathe. They feed over 100 different species directly. They are a lifeline to this entire coast. There's really no substitute for them at all. It's easy to start thinking of DFO as a whole as being somewhat evil, but this is a really good example. In 2004, the year that DFO found ISA in the wild salmon, Dr. Laura Richards, Director of Science, DFO Pacific Region, she's the one that permitted the Atlantic salmon eggs from an Icelandic hatchery that did not meet the Canadian Fish Health Protection regulations. But why did she do this? She said failure to provide permission may trigger a trade challenge by the World Trade Organization. Approval can be granted for such an importation if the risks are deemed to be acceptable. So I think when she says that, she's weighing the risk of this disease against trade sanctions, which I don't know what those risks are. And I don't know if the sanction threat was real, but she seemed to think it was huge. But acceptable to whom? Who was being asked to take the risk here exactly? It was us. We were the ones that got saddled with the risk. And there was an interesting chain of events around all of that. So in 2004, the eggs started to come in. But in 2009, as the missing sockeye were coming through Campbell River, Marine Harvest suddenly began to ask the provincial vet for numerous ISA tests. Rapid fire, 32 requests for ISA testing. Prior to that, they'd only ever asked directly for 2. So this is different from the audits. In the Cohen inquiry, the audit data was there, but also there was a lot of private tests that vet did for the companies, and all of those records are now public as well. In April 2010, the salmon farmers informed the province of BC they're no longer allowed to audit the health of farm salmon. They kicked them off the farms. They said, you can't look at our dead fish anymore. And at the same time, all of these companies stopped importing eggs. Not a single Atlantic salmon egg has come in since 2009, as far as DFO's records go. At the same time as they kicked the province off the farms and they stopped importing eggs, the 3 Norwegian companies signed a memorandum of understanding to share information about viruses with each other, not the government, but with each other. But those farms are on the migration routes of all of our South Coast salmon. And the impact of these viruses on wild salmon is completely unregistered. Nobody is looking at it. Nobody is looking after wild salmon. So this was the biggest thing I found in the Cohen inquiry. There is nobody tasked, nobody with the muscle, nobody with the backup to actually do anything to protect the wild salmon. This suggests to me, this pattern, that there were people who knew that there were big problems with viruses in salmon farms. But I cannot find the response to this on behalf of the wild salmon. I read hundreds of emails, reports, briefings in the Cohen inquiry, and I can report no one in government is allowed to protect wild salmon because DFO has to look after trade, and they have become irrelevant to the guardianship of wild salmon. The province of BC, however, is the landlord to the salmon feedlot industry. They don't even seem to know this. Most MLAs will tell you they have nothing to do with salmon farms. But they're the ones who are renting the seafloor to the farmers who never shovel their manure. They hold all the licenses of occupation that these farms require to bolt themselves to the shore and drop their anchors. They are responsible for the fact that these farms are sitting in the migration routes, releasing viruses into the biggest wild salmon migration routes of Canada. This graph just is something I put together to really clearly present what's going on here. So you have the red line, And that's coming down from Tackle Landing all the way down to Vancouver. And all those red line fish then turn right and go up through the salmon farms. And you can see the graph on the right is what's happening to them. The blue line fish are the Harrison stock. They appear to avoid the farms, and their pattern is the opposite. But they're not the only ones with this pattern. This is a recent graph showing salmon catches. So this is all species for British Columbia. And again, this collapse pattern is evident. If you look at the steelhead for the Broughton Archipelago, where I'm from, very similar pattern again. Timing is all a little different as to when it starts, but once it goes down, these things just bounce along the bottom. This is the Chinook salmon, or spring salmon, from the Broughton Archipelago. This is the South Thompson Chinook. And they're going in the opposite. Now, there's a paper published in 2012, Tucker et al., that talks about these fish, and they appear to have a very similar migration and life history pattern in the early years as those Harrison sockeye. They're found off the west coast, unlike the rest of the lower Fraser Chinook. My feeling is that it's really time to move forward here and use everything we know. I mean, imagine if we used everything we know about wild salmon to bring them back. You know, you look at these fish in the fall, they're starving as they enter these rivers. Some of them go hundreds of kilometers against these rivers. And when they get to their spawning grounds, they blush into reds and golds and greens and stripes, and they build humps and they grow teeth. I mean, they use, at the same time that they're growing eggs and sperm, they devote an enormous amount of energy to wearing this regalia. Why do they do that? They're sending messages to the other fish. And the males and female are looking at each other and going, okay, I have no idea. We have no idea why a female picks a male. You know, what is it about them? I worked at a hatchery for years, and we picked the biggest fish. And we ended up being the only stream in the Broughton Archipelago that coho didn't return to because it was a little stream. It didn't need big fish. So if we let the fish do this naturally on their own, and we used genomic profiling and we sampled these fish at intervals all along their migration route, genomic profiling can see a fish reacting to something long before there's a bleeding sore or it's dead. When they're just the first indications, it's like taking the fish and putting it in the doctor's office and saying, how are you feeling? It can tell you that. It's that powerful. I think it's time to start using these kinds of tools. And I think we're going to have to do it ourselves. I think we're going to have to form the Department of Wild Salmon. And the Department of Wild Salmon, as I see it, will be all of us who are already out there. I mean, there are thousands of people in British Columbia who are working for free to preserve habitat, to restore habitat, to plant eelgrass, to move boulders, to take debris out of streams, to run hatcheries, which I don't think are a great idea, but still enormous amount of energy is put into that for free. There's some kind of extraordinary relationship between people and salmon. I don't know that it's matched by any other animal. Maybe it's because they make themselves so available to us. You can see them, they'll run between your feet. And if we were all to come together, hereditary and elected chiefs, lawyers, biologists, field teams, laboratories, fishermen, elders, the people who depend on the wild salmon, if we formed connections and had a jamboree every year and people came together and we said, you know, hey, our fish are just doing terrible, but these guys like our fish are doing great. And you start looking, okay, what are the differences? And you get together with the powerful scientists who can model these numbers. You produce the data that they need. If you work together and the First Nations and people on the rivers form teams who are out there continuously with the fish in the habitat, checking it, working with the people in the universities who say, you know what, you got to collect the data this way. That's how I did my sea lice research. I had no idea how to study sea lice, as John knows, when I started. But I went back to the scientists that knew what they needed for numbers, and they said, Alex, do it this way. I was like, okay. And do it 3 times, okay. And then you start to understand the trends and see what's going on. We would know almost immediately where these fish were meeting the bottlenecks. And so you wouldn't be randomly saving the salmon. You'd be, okay, you know what? We're going to have to change this, this, and this, because these here, only 10% of the fish are being hit. But here we're losing 90% or 80%. And then you go back to society and you say, you tell them that, and they have to make hard choices. But you would know that when you made a move, when you lifted off the weight of society off of these fish, that they would just flow. Then you're really working, and you'd have to adapt with that. Don't get wired on one method of fixing these fish. Don't build a big organization that has a 10-year plan based on an idea somebody had. You got to adapt. If the fish don't respond to that, stop doing it and do something else. You know, there's two cases where I've seen the fish respond. One is in 2010, where it just amazed everybody, and I watched the coastal communities just turn on. I mean, people were happy, thrilled, thriving. But it also happened in the Broughton. When sea lice first hit, before the trenches were dug and everybody jumped in them and began to defend territory, before that happened, they believed me. And a remarkable man, Dr. Brian Riddle, came forward and he said, okay, let's follow the migration routes of these salmon. And I was like, oh, come on, that's not going to work. You got to take all the farms out. And he said, no, he said, that's what we're going to do. But you pick the route, Alex, because you've been following these little fish around. So I picked the route, they followed it, and it worked. Sea lice fell, and that generation of pink salmon survived better than has ever been recorded in the history of studying pink salmon. But the government scuttled that information. It was hidden. So don't go to government and ask them what to do. Go to them and say, I need a permit for this. But that's the extent of it. We need to come up with the plans. Because over 10,000 years ago, the fish and the people faced a land that was scraped bare by the glaciers. This was a tough place to live for both the people and the fish. But the people respected the fish. There was over 10,000 First Nations living in the Broughton Archipelago. They definitely could have eaten those fish to death. They could have destroyed specific rivers, but they didn't. They had rules and they had ceremonies about letting a certain number of the fish go through. And I don't know exactly how they did it, but they did it, and we can certainly do it again. My sense is that because it's not yet against the law to respect these fish, that we should just take it on ourselves to do it, because I don't think anyone in government is allowed to. There are a lot of good people in the government, there's a lot of good people in DFO, but I don't think they're allowed to protect these fish. And so to continually go to them and beg, which I personally am done with, I will never do that again. I will tell them what needs to be done, I will tell them what I know. And I'm trying now to tell the people of British Columbia about this, because I think if everybody knew this, we could fix it. And we could have a remarkable number of fish. We would not have a salmon aquaculture industry, but I don't think that would be any great loss. The people that work in that industry could easily be absorbed by the wild salmon fisheries and protection, these roving teams, the research. So it's really a win-win situation, but it's going to require us. So I started out studying whales, and it was really fun. And as a little girl, I read every book about people that had gone in the wilderness to study animals, and I noticed a pattern. First, there's the first third of the book is just the great part about them living in the wilderness and all that. Second part of the book is, oh no, the animal I'm studying is about to be wiped out. And the third part was trying to deal with grad students and with the governments around them. And so after 10 years of studying sea lice, and it's still making no progress, I teamed up with two extraordinary people. Anissa Reed is here, and Don Staniford, who's been deported. And I just kept having dinners at my table in Swantula and inviting different brilliant people. And I just said to them all, what should we do next? What should we do next? And finally, there was one dinner where it was just the three of us, and I forget who said it, but somebody said, let's walk down Vancouver Island and make it so that the people of these towns can stand up and say what they really think about the industry. But that is where I began to do activism. And you live 26 years in the wilderness, and you start thinking like a wild animal, and you do not want to be noticed. Wild animals don't want to be noticed. You are then the prey item if you're noticed, generally, unless you're trying to attract a mate. But in this case, you do have to stand up and be noticed, and I started what's called a no, not an organization. SalmonAreSacred.org is nothing. It's a group of people who are amorphous. We come and go. There's the core team. It's now, I don't know how many of us there are, because on any one day it might be 5 or 6. And we've walked down the islands, and we've canoed down the Lower Fraser. 100 people got into 10 canoes that were donated to us. We spent a week paddling down the Lower Fraser in the cold and rain, and every night we were put up in First Nations villages. It was incredibly fun. And then we walked in an absolute deluge to the opening of the Cohen Inquiry, and we walked in with an elk hide that First Nations and others had signed all up and down the Fraser River. It was like a piece of snot by the time we got there, because it was so wet, and we just stood there till Justice Cohen looked at us, and we left, and we gave him one message, and we said, get those disease records out of the farms, and he did. So thank you.